Hello aspirants, welcome to GDC online classes. In this session, we are going to know about the other methods in investigation of biogenetic studies. So up to the previous sessions, we have covered the four methods. They are nothing but use of isolated organ in the biogenetic studies, mutant strain, grafting method and enzymatic method. So all these methods are used to investigate the biogenetic studies with respect to their own specification and own role and own application, because each method will have their unique approach towards investigation of biogenetic studies. If you say use of isolated organ, yes, by the use of isolated organ we can know the site of synthesis. By the grafting method we can know the site of synthesis of secondary metabolites and also the concentration, because we have seen that in the grafting method we have seen that the datura, stalk and tomato scission. Okay, so if we based on that position of these stalk and scission, the concentration of the secondary metabolites also varies, so that each method, so what I'm trying to say is each method have their unique approach towards investigation of biogenetic studies. Clear? Yes, those are all generalized investigation. But apart from that, this method does, namely, you can see right over here on my screen, tracer technique. You know, this technique is a very important and widely employed in almost all branches of science. This tracer technique. So now, today our topic is a tracer technique or a radio tracer technique, or investigation of biogenetic studies by using radioactive isotopes. Clear? Yes. In this session, yes, especially about the tracer techniques we are going to know. Coming to that, the tracer technology now widely employed in all branches of science has its origin in early part of the last century. Yes, from the last century itself it has started, when it was realized the element exists with identical properties but with the different atomic weights. Yes, before understanding how this method will helps to understand or helps to explains about the investigation, but you should know about what is a radioactive isotope. What do you mean by the radioactive isotope or labeled compounds? Which means, yes, in almost all atoms or all molecules are having their identical properties along with some atomic weights, but in some cases the particular molecule is having a same identical property but with a different atomic weight. That should be considered as a isotope. That is considered as a isotope. Yes, in this method, in the tracer technique, we are going to estimate or we are going to investigate the biogenetic studies by using the isotopes. By using isotopes we can investigate. Why we are taking the isotopes? Because these are chemically, they are identical, they are having identical chemical properties, but they differ in the atomic weight. Yes, but these isotopes, in these isotopes also we are having two types. One is a stable and another one is a unstable, which means a radioactive labeled or radioactive isotopes, because the radio which are subjected to the radiation, after they absorb some radiation and they become in almost continuous motion. Okay, they're not stable, they are become unstable, when we subject them into the radiation they become unstable. So while in the unstable form they emit some radiation, so such radiation and such type of compounds will use it to identify, or we use to investigate the biogenetic studies. Yes, I think you got it. But in this radioactive tracer techniques we use isotopes. That too, we use only the radioactive isotopes or unstable isotopes. Clear? Yes, but what do you mean by isotope? Isotope is nothing but which is having identical chemical properties but they differ in the atomic weight. Clear? Yes. For example, the stable isotopes like 2H, 13C, 15N and 18O. So these are stable isotopes. These are stable isotopes, which means they are identically, they are having a chemical properties, identical chemical properties, but they differ in the atomic weight. Yes, when the nucleus may be unstable, such compounds like 1H and 14C, so these are radioactive isotopes. Okay, so because of the uneven molecular weight or maybe some other properties, they decay emission of radiation because they are unstable. The name itself saying that they are unstable. Okay, because of that instability, when we are activating them, they used to emit the radiation. So the property of the isotopes, radioactive isotope, is to emit the radiation. They emit the radiation. Okay, if it is possible to detect these isotopes by suitable means, then they can be incorporated. Yes, because by utilizing these techniques, by utilizing these isotopes, how we can estimate or how we can investigate. Yes, simply we used to incorporate these radioactive isotopes in the presumed precursors. If you simply take an example of a glycoside or a acetate malonate pathway, in that what will be the first step in that acetyl CoA? Okay, so that's a precursor, presumed precursors. Along with this acetyl CoA, if we subject or if we add or if we incorporate any one hydrogen, okay, or 14C, so that will mix up with this presumed precursor, and then it starts and it grows along with it, and it continuously involves in the pathway, it carries in the pathway, and it captures all or the which are intermediates, precursors and enzymes. Okay, like how it is also we need to incorporate the labeled isotope or unstable isotope along with the starting precursor. Okay, so then both will, it will be attached to the precursor, so then it travel along with entire pathway, then it will capture each and everything. Like how I'll simply say that if you, even though you're not understanding, I'll tell you best example. I think all of you heard of a pacemaker which will place in our heart, right? So in some condition, in some cardiac dysfunction, okay, so pacemaker is added. Okay, what is the function of pacemaker? It will examine all the rhythms, all the contraction in each and everything which is involved in the heart. Like how this radioactive isotope is also incorporated inside the stem or root or leaf or anything or seed. Okay, as like the pacemaker, it is also stably, it is also stays inside the plant structure, and it will capture each and everything, how the pathway was carried out and what the intermediates are there. Like how everything gets captured. Like how, as like a pacemaker, this radioactive isotope is also have the functional similarities to capture each and everything in our body. Clear? Yes, I was compared in order to understand how these are acting. Okay, but they are having the unique approach, they are having unique functions with respect to their arrangement and their structure. Okay, yes, because in this simply I want to tell you that, so in this technique we use the radioactive isotopes which are unstable in nature. Okay, so they are nothing but 1H and 14C are the best example. They emit the radiation, so we need to incorporate, we need to place this radioactive labeled isotope along with the precursor, okay, by different methods like stem feeding, root feeding, spraying, and some other methods are there. Okay, yes, and also used as a markers in biogenetic experiment. Yes, biomarkers, they are used as a, sometimes they use a biomarkers. Yes, obviously, even though the biomarkers functions is also investigation of all the functions and metabolic pathways and their key intermediates and all these things. Okay, yes, coming to the definition. Yes, what does it mean? It can be defined as a technique which utilizes a labeled compound to find out, that's what I said in the previous discussion. Okay, labeled compound to find out the trace the different intermediate various steps in the biosynthetic pathways plants at given time and rate. That's what I said. We used to incorporate that particular label compound in order to investigate the different intermediates and various steps. How can you say that the cyclic acid pathway is it is of 7-step mechanism or 7-step pathway? Because someone has investigated by means of some methods like tracer techniques. Yeah, so by this tracer techniques we can know the various steps and the different intermediates and the rate with a given time and rate. Clear? Yes, and also we can define this in this technique the different isotope, mainly radioactive isotope, because that's what I said, radioactive isotopes will decay with emission of radiation, okay, and which are incorporated into presumed precursors in the plant metabolites are used in the biomarker experiments. Yes, that's what is it. And the label compounds can be prepared by two types of isotopes. Yes, so in this you should understand that what do you mean by isotope, and what are the types, and what is the function, and what is the role of these radioactive isotopes in the by investigation of biogenetic studies. Yes, according to that, we are having two types of the isotopes. One is a radioactive isotope and the one is a stable isotope. These isotopes decays with the emission of radiation by they turn into unstable. Okay, they become unstable when we subject them into the radiation. So because and they become unstable form. Okay, so that they emit the radiation. That emission of radiation will helpful for to investigate the and to check the pathway and to observe the pathway inside the plant, because these atoms or these unstable isotopes will emit the radiation throughout the process. Clear? So examples like 1H, examples like H14C, 24 sodium, and also potassium, okay, 42. So these are unstable or radioactive isotopes. When it comes to stable isotopes, this doesn't emit radiation because they are stable one. They doesn't emit radiation and used for labeling of compound. Yes, for labeling of compounds, the stable isotopes are enough. For to investigate the biogenetic pathways, radioactive isotopes or unstable isotopes are useful. Yes, like 2H, 13C, and 15N and 18O. So these are all stable isotopes and these are all unstable forms of radioactive isotopes. So these are helpful for to investigate the all the things, all the key intermediates, all the precursors. But whereas these stable isotopes are to label the compounds, label the intermediates and all the compounds in the all the atoms and all the things in the pathway. Clear? Yes. Coming to that, how or what do you mean by radioactive label compound? The radio label compound means replacement of one or more atoms in a compound by the radioactive isotopes. Yes. In the labeled or stable isotope, if we use it to substitute, yes, here itself it is stable because it is having enough molecular weight. If we add some other, if we add the molecular weight, if we increase the molecular weight, okay, so then it becomes unstable. Yes, and that's what it meant to be. Radio labeled compound means replacement of one or more atoms in the compound by the radioactive isotopes. Yes, in the stable form we used to substitute the radio thing or radioactive isotopes so that it becomes radio labeled compound. Okay, here it is a radio labeled compound. It is radioactive isotope, radio labeled. So stable isotopes are used for labeling and this radioactive isotopes are used for investigation, but we require both. We need to label and we need to investigate. So for that reason, radio labeled compound. So that we need to replace or we need to add one or more atoms into that, stable isotopes that will become radioactive isotopes. Yes, yes. While we are going for this technique, while we are going for to investigate the compound by means of this technique, we need to consider some of the points like the starting concentration of tracer must be sufficient withstand resistance with the dilution in course metabolism. Yes, the starting concentration of the radio labeled compound or the tracer one, like this concentration of these elements will be sufficient to withstand with the further dilution in the course of metabolism. Yes, in the place when we place this radioactive isotope into the plant cycle, so it carries along with the life cycle, it carries all the metabolism, all the metabolic pathways. So meanwhile the metabolism, the plant will undergoes to several changes like the dilution in their metabolism, like somehow the molecules will dissociate and all the protocol will be carried out in such type of dilution things, in such type of diversification, all these things, the tracer cannot be affected, the tracer constant cannot be affected. If at all it is affected, the emission will be lost and also we can't get the product or we can't get that pathway or can't get the such results. Okay, and also proper labeling. For proper labeling, physical and chemical nature of compound must be known. Yes, proper labeling should be there. So in order to do that labeling, a chemical nature and physical nature of compound should be definitely known. Okay, and label compound should involve in the synthesis reaction obviously. because it should involve, it should not be stays at particular starting step, or should not stay at particular point. It should definitely involves in the pathway. Okay, so that only it capture each and everything, it can observe each and everything. What are the key intermediates, what are the precursors, what are the enzymes that are contributing, that will enhance the rate and reaction of the pathway, like how. Okay, and the label column should not damage system individually. Yes, obviously it should be inert. It should involve, but it should not affect. Clear? Yes, these are the criteria for selecting a tracer technique. Clear? Yes, what are the different traces? Yes, my dear students, in the GPAT examination, while we meanwhile we are learning new things, we should consider the points with respect to the GPAT examination, because we are preparing for our goal, that is a GPAT examination, that is especially your primemost goal, GPAT examination. Yes, what I'm trying to say is in tracer techniques, especially from biogenetic studies, sometimes they ask about this type of atoms or tracers for different secondary metabolites, like how they'll give you an option, which one of the molecule or which one of the uh, chemical group or which one of the atoms are responsible for to identify or to use for investigation of such type. Yes, simply they'll give you an option, which one of the following is best to investigate, which one of the following tracer or which one of the following radioactive isotope is suitable for glycoside investigation or terpenoid, like how they'll ask you about specific secondary metabolite, and they'll give you options. In the option they'll ask you like oxygen, nitrogen, and chlorine, like how I'm just saying. Okay, so for that reason we can, maybe there is a chance we can get the questions from this tracer technique also, like how they'll give you a specific secondary metabolite glycoside, and they'll ask for the specific tracer compound or radio labeled compound or radio labeled atom for the investigation of such secondary metabolite. And for terpenoids, oxygen atoms are enough. Okay, for glycoside, oxygen, nitrogen, okay, sulfur and carbon are required. Okay, so for studies on protein, alkaloids, amino acids, labeled nitrogen atom will give the specific information. Yes, they'll ask you like here also. Okay, as like here also they'll ask, they'll give a direct secondary metabolites. So for alkaloids, which tracer compound or which labeled compound is enough or required? Nitrogen, sulfur, and like how they'll give you an options. Okay, please remember the compounds which are specific to that secondary metabolites. Yes, up to now we have seen what is meant by the radioactive isotopes tracer techniques and the criteria and types of radioactive isotopes, and we have seen some of the examples. Okay, yes, coming to that, we need to know about what are the how the tracer techniques will be carried and how these are performed. So nothing but steps involved in the tracer technique. First one is selection of radioactive isotopes. Yes, why I have placed different atoms for the glycoside, why I have placed oxygen for terpenoid, why the nitrogen is required for the protein and alkaloids and amino acids? Simple, with respect to their chemical composition. Okay, because we are having a specific atoms. Simple example, I'll tell you that nitrogen is used to investigate the protein, alkaloid, amino acids. Can anybody tell me what is the link between this nitrogen and this amino acids and alkaloids and proteins? Because the amino acids are nothing but having nitrogen, the building block. Alkaloids having a nitrogen containing heterocyclic ring. Yes, and proteins are made up of amino acids. So for that reason, as like this, every compound or every secondary metabolites having a specific radio labeled compound. So for that reason, with respect to perform biogenetic investigation of biogenetic studies by means of tracer techniques. Okay, first and foremostly we should select a radioactive isotope with respect to the secondary metabolite. As like if you want to investigate glycoside, you can take any one of like oxygen, nitrogen, sulfur and carbon. Clear? Yes, with respect. So first you need to decide which compound you are focusing, which thing you are going to investigate. First you need to decide, then after that you should select the radioactive isotope. Okay, like how? So they should have a long, and also after deciding you should select that. Among that we should consider some of the points like the particular radioactive isotope should have a long half-life, and they can continuously show their radioactivity throughout the investigation of study. Yes, that's what I said. It should be stable, it should involve, okay, and it should emit the radiation throughout the process, so that only we can capture each and everything. Okay, so it should have a long half-life, and they can continuously show the radioactivity. Clear? Yes. And the selection of suitable isotopes based on the chemical composition of sample. Yes, that's what I said. With respect to the compound that we are going to investigate, accordingly we should select the second radioactive isotope. Clear? Yes. It's coming to that. Considering the half-life, the 14C has a half-life of around 6,000 years. 6,000 years, so that you can assume, you can imagine. So because of this half-life, the carbon is the most preferable one, and also we can investigate each and every plant with respect from the starting plant to higher plants, like very old plants up to several thousand years. Clear? Yes. So investigate, so that we can by this point you should understand that we can investigate even though it is having a several years. Like how? Because the 14C having a 6,000 years half-life, so that we can have this, or we can by this we can investigate even though the 6,000 years old plant or old tree. Clear? So that's the beauty. Next one, after selection we should prepare a label compound. Preparation of label compound. Yes, this will see how the preparation will be done. Label C compound can be prepared by growing in a chlorella in carbon dioxide environment, especially 14 carbon containing carbon dioxide environment. So in which we place the label compound in this environment. Okay, so but what do you mean by chlorella? Chlorella is a green algae. Okay, when it grown in C14 carbon dioxide environment, all C compounds are 14C label. 14C label. Yes, if you want to label, if for assume, okay, for example, assume that I'm going to label carbon. Okay, so that I should select an environment which is rich in carbon dioxide, especially with the 14C carbons, and that too they should be in a chlorella environment. They should be grown in chlorella environment, because it is a green algae. Okay, so when we place the label carbon into this environment which is consisting of chlorella algae, which is having a carbon dioxide, especially 14C carbon dioxide, when we place them and we cultivate them, finally the compounds all are that we incorporate, that we insert, all become the 14C carbons. Okay, yes. So those can be further isolated. Yes, after that they can be isolated. They are directly incorporated for further processing. Yes, the tritium 3H compound are commercially available. Yes, but so this environment here, for to label a carbon compound, we should prepare this environment, but where in the hydrogen itself we are having a commercial varieties. 3H is a tritium, is a commercial compound is available in the market for labeling of such compounds. Okay, the tritium labeling is done by catalytically exchange in aqueous media by hydrogenation of unsaturated compounds with tritium gas. Simply we used to place the tritium 3H, okay, the tritium gas. In this we used to place the compound or the thing which need to be labeled. So when we place it in that particular gas, so obviously the catalytical exchange will be done in the aqueous solution or aqueous media, so that the compound become labeled. Okay, yes. Next one is incorporation of a label compound into system. First we have selected the isotope, then we have prepared the label compound, then it is ready. So we need to incorporate, or we need to insert, we need to feed into the life cycle, or we need to feed into the plant. So for that reason we are having a incorporation of label compound into system. Okay, so first one is root feeding. Simply with nothing but we need to feed, or we need to incorporate from the root itself. So plants are cultivated hypotonically to avoid the microbial contamination. In the method, label compound is mixed up and incorporated along with the nutrients and minerals, and hydroponical cultivation. Yes, hydroponical cultivation in simply nothing but we never use any of the soil, but hydroponically we cultivate the plants meanwhile in the cultivation, simply from the root itself, so just simply we'll prepare, okay, we prepare, we place the water and other nutrients in the, okay, other nutrients. So in that water and nutrient media, we used to place the particular compound, okay, particular radio label compound. Clear? I think you got it. Simply as like the nutrient, as like the fertilizers, as like the other things which we supply, okay, not on the soil but we supply by means of hydroponical cultivation. Clear? Yes, to prevent the microbial contamination. If it is in the, if it is attached to the soil, if it is to the land, it may be contaminated. So for that reason, one method is root feeding. Simply directly we used to supply by means of root, simply as like we place the other water and all these things. Next is stem feeding. Stem feeding, it's another method of incorporation of the compound. In this, we used to see substrate can be administered through the cut stems immersed in a solution containing radioactive isotopes, and latex containing plants are exceptional for this type of feeding. Yes, simple. In the root feeding itself, we supply the direct radioactive isotopes along with some water and all these things. In stem cutting and stem feeding, we used to isolate the stem, and we need to place it in a water on other nutrient which is having a already having the radioactive isotope. Okay, so simply we need to immerse. But for this stem feeding, the latex releasing stems are exceptional because they won't absorb the radioactive isotope or nutrients. Instead of that, they release some latex. So for this thing, they are exceptional. Okay. Yes, then direct injection, simply either maybe for from the root or stem or maybe other parts, or for fruits, we used to inject direct injection of the radioactive radio label compounds. Okay, so mostly it will be done for umbelliferous fruits and capsular fruits. Okay, yes, so direct injection. And floating method, in this plant method, is allowed to float on the solution containing radioactive isotopes along with some nutrient media. Almost all, okay, it should supply along with nutrient media and water, but the mode of application will be varied. If it is root feeding, simply we'll directly place on the root. If it's stem feeding, we cut the stem and we should place it into a nutrient and radioactive isotope containing solution. And direct injection, we used to inject to the stem and fruits. And whereas the floating method, we need to place a plant on the floating solution containing radioactive isotope. And finally, spraying method. Simply we used to spray wherever we want to investigate. So we simply spray the solution or the radioactive isotope. So these are the methods to incorporate. So up to now what we have seen: selection of radioactive isotope, preparation of label compound, and incorporation. So we have incorporated. So up to that, imagine. Okay. So we have incorporated. So what you should do after that, you should do its own function to highlight and to give some of the ideas regarding investigation or the pathways. Okay, we'll see what is the next one. Separation or isolation of label compound. Yes, we have selected, we have prepared, and we have incorporated. And after that it has traveled into the plant life cycle, and it has captured, it has detected the intermediate precursors and all the end products. But after that, we need to separate or isolate the label compound in order to check what things it captures and what things it has noted down. Yeah. So separation or isolation of label compounds. The separation may be done based on the nature of sample plants, like plant parts like soft fresh tissue, hard tissue, fractional crystallization, and partition chromatography are used. So these are the methods to separate from the plant part. So the plant part is having radioactive isotopes. Okay, but if it is a soft tissue, we can separate the radio-labeled isotope, okay, label compound, by means of infusion method, maceration. For a hard tissues, we need to go for percolation and decoction, and we can separate by means of fractional crystallization and partition chromatography are used. So this is how we separate. But we need to detect the compounds even though it has traveled along with all the precursors. But how can you say that what type of precursor, what type of things are there? Okay, in order to know that, we should detect the compounds. For this detection of compounds, we should have a technique called autoradiography. In this technique used for location of radioactive isotopes in biological and other material. So in this method, what in order to check the location of radioactive isotopes in biological and other methods, we need to check the autoradiography. In this, specimen is placed in contact with suitable emulsion, nothing but X-ray sensitive film. Okay, and after that exposure to the latter is developed in the usual manner. Yes, and the resulting autoradiograph gives a distribution pattern of radioactive substance in the specimen. Then this method can be applied for all morphological plants or histological sections. For its resulting negative is used under microscope. Yes, obviously. So to check if it is a plant, entire plant. Okay, assume it is an entire plant. Okay, it's the roots. Okay, and there's a root tip, sorry, shoot tip. Okay, yes, we don't know whether it is, where it is, not whether it is. Okay, we don't know. I have incorporated by means of a roots feeding. Even though it is traveled, the plant has grown. But how can you say that at which location this radiolabeled compound is say there? Yes, to that we have placed that autoradiography. Okay, in that we need to subject this particular thing into a suitable emulsion. Then after that is subjecting to the radiation, so that the distribution pattern of the radioactive substances will be identified. Okay, I place entire a leaf or shoot or everything. Okay, so if anything which will shows a emission or any of the distribution pattern of radioactive substance will be observed, so at that period, at that location, the radioactive isotope is there. Okay, yes, nothing but it's to detect the compounds, detection of radiolabeled compounds. So simply autoradiography. In this autoradiography, we used to know about the location of the biological and other materials or radiolabeled isotopes. Okay, in that we need to place it in a stable emulsion after the subject into radiation. Okay, we need to examine. So if it releases some emission, that will be the release pattern will be changed. So somewhere if it release pattern will be, or distribution pattern will be varied, so that you can assume so there is something or there is a radiolabeled isotope. So that's one method. Next is Geiger-Müller counter technique. Yes, it's a tube. Geiger-Müller tube is a Geiger-Müller tube. So in this tube there is a device which is used to detection of measurement of all types of radiation, alpha radiation, beta radiation, gamma radiation especially. Okay, but basically it's consists of two pair of electrodes and surrounded by a gas. The electrodes have a high voltage across them. The gas used initially is a helium or argon. When the radiation enters into the tube, it can ionize the gas. The ions are attracted towards the electrode and electric current is produced. Simple as like the radiation, as like the spectroscopy. Simply here it is also very similar. Okay, but here we place electrodes. Positive and negative electrodes are there. Okay, it is just surrounded by gas. The gas is nothing but helium or argon. You should remember that. Which gas is used in Geiger-Müller technique? You should remember these two gases, helium and argon. When you place a particular thing called the compound into that, after the extracting into that, we need to place in a Geiger-Müller tube. It is having two electrodes which is surrounded by the gas. Okay, so when the electrodes have a high voltage across them, they usually having fluctuations with respect to the radiation emitted by the compound, so that we can detect as like a normal spectroscopy absorption or radiation. Here itself we can observe which type of radiation is there, alpha or beta or gamma radiation. Clear? Yes, that's about the Geiger-Müller counter technique. The next one is scintillation counter. Next one is scintillation counter. Nothing but scintillation counter consists of scintillator and device such as PMT, simply photo multiplier tubes. We can see these detectors. The scintillator consisting of PMT. PMT is a detector. Photo multiplier tube detectors which we see in the mass spectroscopy and all these things. Okay. Well, what happens in this? When we place in order to, for these techniques all are for to detect the label compound. Yes, in this simply we place in the scintillator tube. The label compound is placed in scintillator tube. Okay, so it is connected, which is having PMT as a detector. Okay, so when we place a label compound, when we subject or when we increase the radiation, okay, electrical signal, the light converts into electrical signal, and the scintillator is a material that exhibits the scintillation. Okay, and the property of luminance when excited by the ionizing radiation, and there's nothing but simply the light into electrical signal. Yes, obviously when we place some light in the scintillator, the light energy is converted into electrical energy or electrical signal. So based on the signal we used to detect the compound, or based on the peaks we used to detect the compounds. Clear? Yes, so this is about the three techniques are for the detection of isotopes. Yes, coming to that, we'll see the last thing, types of tracer techniques. Methods of tracer techniques. Yes, this is also important one while we are discussing with respect to the investigation of biogenetic studies. Okay, so up to now we have seen what is a biogenetic, what is a tracer technique, what do you mean by say radioactive compound or unstable compound, the criteria, and also the steps involved in the uh, investigation of radioactive isotopes. Okay, yes, but what are the methods for tracer techniques? What are the methods for tracer techniques? Yes, coming to that, precursor product sequence. Precursor product sequence. So we can perform the tracer technique based on to target the both precursor and product sequence also. Okay, so yes, this method is for to use and for the illustration of biosynthetic pathways in plants by means of label compounds. By means of label compounds. Yes, how can you say, you know, by nothing but starting precursors like 6 carbon dioxide molecules, 6 H2O molecules. How can you say that's the precursor? Those are starting products because of like some techniques, precursor product sequence. So this method allows you to understand or allows you to demonstrate that this precursor will lead to a product. Precursor and product sequence. Precursor and product sequence, so that we can understand what is a precursor and what product is formed. So starting and end product can be assessed, or such type of, you know, if you want to perform, if you want to investigate product and precursor and product sequence, you should perform this. Okay, so the precursor and product sequence is commonly invoked in this. A presumed precursor of a constituent under investigation is a labeled form, is fed into plant, and after suitable time the constituent is isolated and purified, its radioactivity is determined. Clear? It's like a normal protocol. Okay, so we need to incorporate the presumed precursor, so and also we need to label it, and after that it should be investigated for the other things. Yeah, so based on their radioactivity. So there's a normal technique as like we explained in the previous one. Okay, but here itself the radioactivity disadvantage is having, it is isolated. The radioactive isolated compound alone is not usually sufficient evidence that the particular compound is fed in direct precursor. Yes, the radioactivity of isolated compound, so that we have isolated now the thing which is used for a compound. Okay, so that radioactivity is not enough, because maybe the same radioactivity will be having for some other compound. So for that reason, in this product precursor product sequence may be having such type of disadvantages, because substances may enter into general metabolic pathways of the plant. From there they become randomly distributed through whole range of products. Yes, obvious, that's what they meant be. Okay, so the isolated compound radioactivity cannot be assessed, because the isolated compound previously it have gone so many changes. Okay, so that we cannot assess whether it is the compound, whether this isolated compounds is a thing which emits all the radiation that we have captured, like how. Okay, but coming to that, you should remember that what is which method is for which thing. For a GPAT examination, you should remember that. So by which method of tracer technique we can assess the biogenesis of morphine and 𝘿𝙄𝙎𝑎𝑑, precursor and product sequence. So by this method we can know the biogenesis of morphine and 𝘿𝙄𝙎𝑎𝑑s. Yeah, this is one method. Another one is a double and multiple labeling. In this thing, one scientist has done this. Okay, 𝘿𝙄𝙎, yes, nothing but 𝘿𝙄𝙎. He has done an experiment that he just want to know which one or which compound, which label atom will helpful or leads to formation of end product or formation of the secondary metabolite. So for the reason he has performed an experiment, you know, two doubly labeled lysines to determine which hydrogen is of the lysine molecule was involved in the formation of pi-pyridine ring. The pi-pyridine ring. Okay, so which hydrogen molecule? So lysine, this is a lysine molecule. Okay, lysine compounds. From this, which hydrogen, okay, which hydrogen is responsible for the formation of pi-pyridine ring? Okay, yes, by that he had done that multiple labeling. So at here itself, and here itself, he observed that he labeled at different positions in order to understand which label compound is responsible for which formation of pi-pyridine ring. So that by this method, simply I'll say that by the double and multiple labeling method, we can assess which label compound is responsible for formation of end product. Okay, so like how we we do the procedure. So this method gives evidence of natural biochemical incorporation of precursors, erases double and triple labeling. Okay, yes, in this method specifically labeled precursor and their subsequent degradation of recovered products are more employed. That's what nothing but here we displayed. Okay, so it is observed in anabasine and nicotine. So this is another method. And last but not least, a competitive feeding. It is nothing but in this competitive feeding, you can have a look on this image. Okay, yes, in this particular method, each method having their unique way of approach or the way they are having. Okay, nothing but simply in one word I'll tell that in this method, in this competitive feeding tracer technique, what we know or what we can get from this thing, if you ask me like that, sir, what we can what is speciality of this competitive feeding method, simply I'll tell you that we can see the what are the possible intermediates to form this end product. So that's what it meant to be. Yes, there can be value in determining which of the two possible intermediates are normally used by the plant. So the normal intermediates are used by the plant. Okay, so this is what meant by the competitive feeding. This is also one of the tracer technique, but by this method we just we can know that for formation of such end product, what are the key intermediates, what are the two possible intermediates, so we can assess. So that if you I'll tell you that C6H12O6, that is a glucose. What are the possible intermediates or what are the possible precursors? 6 carbon dioxide molecules and 6 water molecules. Okay, like how these are two possible intermediates only to form a glucose. Like how it can assess the two possible intermediates for the formation of the end products. Okay, especially this method is to use for elucidation of biogenesis of propane alkaloids. Illustration of propane alkaloids. Yes, and next one is sequential analysis. In this, a second method of investigation with C14 to grow plants in atmosphere of, yes, obviously as like a green algae, precursor product sequence, as like the same scenario here also, but within a time intervals to obtain the sequence in which various related compounds become labeled. Okay, the precursor is the precursor product sequence. We do the same procedure, but here with the time intervals we do the procedure to obtain the sequence in which various related compounds become labeled. Okay, yes, for this thing applications are like, so what we can get, this method has been very successfully used in illustration of path of carbon in photosynthesis. Yes, also for determining the sequential formation of opium, hemlock and tobacco alkaloids. Should remember all the compounds which are investigated, so that will be enough good enough to answer all the questions that we are expecting from. Okay, yeah, so that we can get, as like I said, that photosynthesis. So yes, my dear students, this is about the investigation of biogenetic studies by means of radioactive isotopes or stable isotope, unstable radio unstable compounds. Clear? Yes. Please go through with a video. If you're having any doubts, please have a look on all the content which I have provided in this slides and this video, so that you can understand. Clear? Yes. First, more such content, do install our app and do follow our YouTube channel, that is GDC English classes. Clear? Yes. Thank you, students. Have a great day. We'll see you in the next session.